Hi everyone, welcome to my talk. My name is Debbie Winter, everyone calls me Debbie. I'm just finishing up my PhD at Duke University in computational biology. Today I'm going to tell you about how we use DNase-seq to annotate stable nucleosome positions across the human genome. Nucleosomes, as you probably know, are interesting because they play a role in gene regulation by controlling access to regulatory regions and transcription factor binding sites. Now, at this point, you might be asking yourself, can't you just do this with MNase? Yeah, MNase is a similar assay where an enzyme digests the linker region between nucleosomes. Then mononucleosomes are isolated, and the associated DNA is sequenced. Typically, the location of the dyad, or center of the nucleosome, is inferred from the end of overlapping sequences. For this reason, MNase is really good at finding well-positioned nucleosomes, but it can overlook nucleosomes classified as fuzzy or shifting across the DNA in a population of cells in a sample. They can be overlooked because their ends don't align exactly. Moreover, DNase can be used to ascertain translational position or the general sequence covered by the nucleosome, but the exact position of the dyad is uncertain. This means that it's hard to determine rotational positioning or how the DNA is oriented on the histone. So how is DNase-seq different? DNase-seq is an assay commonly used to identify open chromatin, regions that are nucleosome depleted. But in our DNase protocol, we find that about 2/3 of the sequences actually fall outside these DNase hypersensitive regions, or DHS. It's these sequences, those that are in the nucleosome-occupied regions, that we're interested in. In this figure, we recorded the distance between adjacent DNase-seq reads, either mapping within DHS sites, the purple line, or outside of DHS sites, the blue line. The number of reads on the y-axis and the spacing between them is on the x. We find that the spacing in non-DHS reads appears to oscillate, as you see, while the DHS regions show no detectable periodic pattern. This period is about 10.4 bases and coincides with the length of one turn of the DNA helix. This pattern has actually been known for a long time. It was originally seen in gel electrophoresis of mononucleosome fragments digested by DNase. Here we see it observable on a global scale and when we were actually looking at 49 combined cell lines. Since this pattern is not visible in the nucleosome-depleted DHS sites, we believe that it's related to nucleosome positioning. All right, so here's how we think it works. We know two things about DNase digestion. First, the enzyme cuts in the minor groove. And second, it cuts with a 3' overhang. So when DNA is unbound, like in a DHS site, DNase is free to access the DNA from any angle. It can cut anywhere, and you get the pileup of reads. On the other hand, when DNA is bound to a histone in the nucleosome, DNase can only cut in the optimal orientation once every turn. And this is what leads to the 10 base pair pattern. Moreover, the 3' overhang means that the digestion site between strands can vary up to 3 base pairs. The data we used for our study actually come from 49 cell lines, which we combined to increase the density of reads in these regions. The fact that we still see these patterns even across cell lines means that the rotational position is being conserved. So either this means the nucleosomes are in the exact same position in every cell line, or more likely, and supported by the literature, the histone is still sliding in these 10 base pair increments so that it maintains its rotational preferences. Now that we know this expected pattern of the DNase digestion around the nucleosome, we want to use it to reverse engineer the nucleosome positions. So here's kind of our pipeline for calling what we've termed DARNs, or DNase annotated regions of nucleosome stability. We start off with our read counts from 49 cell lines, which is about 15 billion data points. Then we use our empirically derived pattern of DNase digestion, shown here, and we slide it along the genome looking for good nucleosome matches. We do this by calculating the correlation between our pattern and the reads around each base in the genome. Then we end up with this purple track, the correlation scores. From these, we define peaks, or the best local match, for both the positive and negative strands. These peaks are the input for our hidden Markov model, or HMM, that distinguishes from background regions in the nucleosome state, which means that the expected periodicity between correlation peaks on the same strand and on different strands matches what we think it should look like. From this, we end up with about 14 million DARNs. Next, we were interested in how our DARNs overlapped other nucleosome annotations. we first looked at in vitro nucleosomes where naked DNA is combined with histones. Then the Manase protocol is performed and well-positioned dyads are called. When we plot the distribution of the in vitro dyads relative to the midpoint of the nearest darns, we found that they were enriched on either side of the darns, so here and here. We believe that these dual peaks mean that the darns calls are actually falling on either side of the dyad, so you can kind of see what I mean here. The same is true when we look at the results of an in vivo Manase experiment in GM12878. In this case, we're plotting the nucleosome occupancy signal, and it's relative to the darn's midpoint. This seems to imply that the periodicity is lost or out of sync over the dyad. Now, since many of our darns seem to be covering one side or the other of the nucleosome, we're going to use this opportunity to look at the differences between the halves. All right, so we annotated a subset of darns as either 5' end left or 3' end right of the in vitro dyads. Then we plotted the DNase read counts centered on the midmost peak of the darns. First, we're looking here at 5' end darns. We note that the periodicity of reads, which is expected based on our model, of course, delineates the different portions of the plot. We see the strongest signal over the part of the nucleosome covered by our darns and the weakest over the linker. Now, when you look at the 3' end darns, now on the right, you see that they're essentially reflected with the linker now on the right. When we do a similar analysis with the A and T base frequency relative to the darn's midpeak, we see the same trends essentially. We note that AT are depleted in the nucleosome area, which is consistent with CG-rich nucleosome cores and AT-rich blanks. We also find that the periodicity of A's and T's is in sync over the region of the nucleosome covered by the darns and falls out of sync over the dyad and through the linker. Moreover, the AT signal is out of phase with the peaks in the darns. Since DNase cuts in the minor groove, as I said, this is consistent with the literature contending that AT dinucleotides align to the major groove of nucleosomes. We were also interested in how our darns were placed around genomic features that are known to act as boundary elements in positioning nucleosomes. When we map the distribution of darns around promoters, so here the blue line with 0 being the TSS, we find that they're depleted for darns relative to random introgenic sites, and you might say that you see a bit of an enrichment here downstream towards the gene body. Similarly, when we plot darns around the midpoint of DNase hypersensitive sites, looking at ubiquitous ones, those that are found in all cell lines in green, we see a marked depletion. On the other hand, when we look at all DHS, which includes mostly cell type specific, in the orange line, the darns appear to have shifted towards the center. So we're looking at ubiquitous versus all. So since we have complete DHS data for all the 49 cell lines in our study, we decided to look further into cell-specific effects of darns. In addition to our data, we also ran the darns HMM pipeline on a combination of 70 lymphoblast cell lines, or LCLs, that we retrieved from the Pritchard lab. When we compare these darns, we end up with 3 sets. We have the 49 specific, those that were only found in our darns, the LCL specific, those found only in the Pritchard data, and the shared, those that were picked up by both. You can think of the 49 as being ubiquitous since they come from diverse cell lines, whereas the LCLs are our cell-specific data. In these figures, we plot the distribution, just like before, of different sets of darns around either DHS that were found only in LCL, LCL specific, or those found in non-LCL lines. We see that the results conform to our expectations. For example, if you focus on the one for the LCL specific darns, you find that they're depleted for they're depleted for darns in the LCL specific, where we expect them to be open, and actually enriched for darns in the non-LCLs that we would expect to be closed in the LCL line. All right, to summarize what I've shown you today, DNase can annotate regions of rotational nucleosome stability. These features are conserved across 49 cell lines, and we validated them in the LCL data set. They also could have implications on transcription factor binding. For example, if some transcription factors have a preference for binding a certain DNA orientation, their motif position relative to the minor groove could provide clues as to where that might be. We also show that darns tend to fall on either side of the dyad, And I explained earlier that this could suggest a loss of periodicity at the dyad of the nucleosome, and that would mean the DARNs might even map to the same nucleosome on either side. Conversely, another theory could be that the nucleosomes are being alternately exposed to digestion. I also showed that DARNs border genomic elements in a cell-specific manner. And finally, we're planning on continuing our work by looking into the role histone modifications play in these rotationally stable nucleosomes, and also what the DARNs might tell us about higher order structures in the chromatin. Thank you for your time, and I'd like to thank the RECOMB organizers for giving me the opportunity to share my research. I'd also like to express my gratitude to my advisors, Greg and Terry, as well as the various lab members who offered me guidance and support. My PhD work has been funded by NSERC, and the lab has been funded by ENCODE. Thank you.
Okay, maybe I'll start with a question. So I'm curious, how hard is the protocol in terms of trying to apply it to in vivo tissue?
I'm sorry?
If you wanted to apply the protocol to in vivo tissue, could you do that?
So the studies that we've been doing, like the 49 cell lines are all in vivo. Is that what you mean?
I mean, if you collect primary tissue from an animal. And you
Okay. Yeah, I mean, in theory that it could be done. Right now, they're working on making DNase be able to be used in tissues. Most of it has been done on cell lines and loose cells. But in theory, it would work perfectly fine. In theory, you could do it with really deep sequencing. You could do it on a single cell line, and you could get positions in a single cell line. It's just that at the time that these experiments were done, it made sense to combine them.
Okay. Okay. If there are no more questions, let's thank the speaker again.